Welcome to the MVP, the ModernVivo podcast. I'm your host, Andrés Velarde, Director of Operations at ModernVivo. And in each episode, I sit down with a different life scientist to talk about their research findings, the implications of those findings, as well as their career trajectories and the steps they took to arrive where they are now. Today I have a discussion with Dr. Georgia Lattanzi, cancer immunologist and researcher at the Salk Institute in San Diego. We discussed their research paper published in Mucosal Immunology that looks at the crosstalk between invariant natural killer T cells and neutrophils and what that means for people with colorectal cancer. Stay tuned to learn more. All right, Dr. Georgia Lattanzi, welcome to the ModernVivo podcast. How are you today?
Good. Thank you.
Yeah, I'm really excited to talk with you about immunology, which has been a key focus in your career. And before we get into it, do you want to just tell our listeners who you are and what you're working on these days?
Yeah. Well, first of all, thanks for the invitation. And I'm always excited to talk about science, about immunology. And so thanks for this invitation. So I'm a postdoctoral fellow at the Salk Institute for Biological Studies in San Diego. And I'm an immunologist. Specifically, I'm a cancer immunologist. That means I've always studied the immune system in the context of cancer. Yeah, and specifically, so basically, the immune system is the body surveillance system, right? And it is made up of two different arms, the adaptive and the innate immune system. And my expertise reside at the connection, at the interface between the two. So I've always studied cells that bridge the communication and the direct the responses of both the innate and the adaptive immune system.
Excellent. Yeah. And for context, I found you because I was looking for papers on CAR T cell therapies, which is like very popular, it has been for a while. And then I found your paper published in 2023 in Mucosal Immunology called iNKT cell neutrophil crosstalk promotes colorectal cancer pathogenesis. Can you tell me before we get into the details, like how did you come to this work, or at a high level, what was the investigation about?
Yeah, so this work is the work that came out of my PhD project. And basically, we started from a really simple question. We wanted to study this unique population of immune cells that are the invariant natural killer T cells,
Mm,
iNKT. And in the lab where I was working, which is the lab led by Dr. Federica Facciotti, we were interested in studying these cells because they are so peculiar. So basically, they are really rare. So you think like, oh, they're like about 1% of the immune cells in the spleen, in the liver, in the lymph nodes. So you
yeah.
think, why are they still kept and maintained, and what do they do? So it's really a matter of quantity or quality.
Mm,
Actually, it's quality, right? And I mean, we just started because the iNKT cells, they do, one of their characteristics is that they respond to glycolipid, so antigens of a lipidic origin. And the intestine is an organ where you do have many lipids that could come from bacteria, from food. So we started to work from there. And they are really important because their characteristic is also that they can react strongly and fastly. So the iNKT cells really represents a good platform for cancer immunotherapy. So we
mhm.
need to study them more.
Yeah, certainly. And just based on my kind of first read or second read through the paper, it seems like the main takeaway is that like the microenvironment and the microbiome really influence how the iNKT cells behave. Is that correct? So they're not always good, always bad, but it really switches like in terms of the phenotype. Walk us through, how did you like make that discovery?
Yeah, exactly. That's actually one of the main takeaway from the paper is that they can be fastly and readily shaped, and then they are strong enough to shape an immune response that can come after their activity.
Hmm.
And yeah, so Actually, the approach we had for this paper was to start from human patients. So the idea, because iNKT are so rare, they are really not well studied, there are not many studies in human. So our first question was, can we see them in humans? Are they
Mm.
present in tumors? So we really started by having an eye on the human side. And to do that, we established a really fruitful collaboration with the clinical scientists, so with oncologists, with physicians, surgeons, and we collected many, many samples. We collected like the tumor samples fresh, which is really precious,
Hmm.
and then the non-tumoral adjacent side of the tissue, and then the bacterial component, the mucosal component of each tumor. So we collected as much as we could, let's say, and and then we did a strong and deep immunophenotypization of these tissues. And yeah, the first big finding was that we were able to identify and to detect them. And we could observe that there was an increased infiltration of iNKT cells in the tumor
Mm.
compared to the adjacent tissue. And then we obviously looked at all the other factors, like, you know, other immune cells in the environment, cytokines that were generally produced, the bacterial component. And we then did a lot of correlation analysis to
Hmm.
see whether some specific features of the tumors could be associated with the presence of the iNKT cells and with the phenotype they had in the tumor, which was different than the one they had outside of the tumor. And yeah, we found out that they did correlate with the presence of neutrophils, actually pro-tumor neutrophils. And we also observed that they could be, that their functionality could be skewed by some specific bacteria that we
Mm.
observed in our patient cohort and that are known to be like oncogenic, like drivers of tumors.
Yeah, fascinating. And it sounds like you've also used like an in vitro and in vivo approach to corroborate some of your findings. So yeah, I'm curious, just like how did the chain of logic go, right? Like, did you like collect all these samples and then did you see the result immediately, or did you have to keep collecting and then you decided, okay, let's do an in vitro test to see if these signals are actually real? Or can you kind of walk me through the different decision
Yeah.
points?
Yeah, so we started, as I said, from the human, and we had to collect a lot of samples at first because also human samples are quite heterogeneous, right?
Hmm.
And once we started having a clear picture of what was the phenotype and basically what's the phase of these cells. We then decided to move in vivo because then the next question was more like, okay, so what's their functionalities? That's what they are, but like, what are they actually doing? And can we intervene? Can we do something and see? And that's why we then moved in vivo. And since I believe, and in the lab, I mean, science is, you cannot tackle one question just in one way.
Yeah.
So my boss really was really, she was really like highlighting that we had to have different models to see whether we could get to the same answer in
Right.
different ways, because that's really strengthening your finding, right? So we did establish like three different models of in vivo tumorigenesis of colon cancer. And we were lucky enough, I mean, lucky, we could observe the same phenotype that we have seen in the patients, which really made us believe that that's the phenotype associated with iNKT cells in tumor cancers. And
Yeah.
again, just inside the cancer, because outside they would have a different phenotype. And one of these models, for example, was a really like a microsurgical model that I went to Switzerland to learn how to do because it is a technique that it needs a lot of training and it's
Yeah.
not something that is usually done. So it's basically orthotopic implantation of tumor cells in the intestine. So it's a microsurgery. I would call myself a mouse medical doctor.
Yeah. Wow. Yeah, I bet. Wow. It sounds really precise. Interesting. So I'll certainly make the paper available for people who want to read more because there's
Yeah.
so many details that I feel like we won't have time to go into. But if you were to zoom out and think about like the greater implications of this finding, what does this mean for people with, colorectal cancer?
Yeah, the greater implication, I think, is that we highlighted and we identified a pathway that could be tackled for therapeutic intervention, which is the usage of iNKT cells in cell therapy. But our findings is highlighting that you cannot just think about the therapy per se, but you have to take into account the tumor microenvironment.
Mhm.
So, it's not just going to be iNKT cell therapy like T cell therapy, but you need to think about the microenvironment, in this case, the bacterial microenvironment, because it might influence your cells and then, shape their action. But at the same time, you can act directly on the bacterial content of these patients with different type of treatments to actually improve different outcomes.
Yeah.
Because, because also the main finding of this paper is basically that the iNKT cells are shaped by some oncobacteria, specifically Fusobacterium nucleatum, and they acquire a protumorigenic phenotype. So they kind of sustain tumor formation. And
Mhm.
that's nice. That's nice. I mean, that's really corroborated by in vivo analysis in which we use also knockout animals that lack iNKT cells. And again, we use two different models, two different genetic models of animals that lack iNKT cells. And in both of them, we observe that lack of iNKT cells is associated with a lower tumor formation. And then we also observed, going back to our clinical patient cohort, that an increased infiltration of the specific phenotype of iNKT cells in our patients would lead to lower survival probability.
Yeah. Yeah, thank you for sharing. I mean, really, really important work. And I'm curious how this translated into your current work. So last we spoke, it sounded like you were kind of moving away from colon cancer into more like breast cancer and B cell investigations. Can you tell us more about that work?
Yeah, so I'm still a cancer immunologist. I think that's exactly what I am. And after my PhD, I transitioned into this postdoctoral position. In the lab where I'm right now, we study B cells, which are again cells that are, that crosstalk between the innate and the adaptive immunity. So I think I really like th- th- this positioning of the immune system in being able to act and direct different immune responses. And I studied a role in solid cancer, specifically breast cancer and head and neck cancer also. Uh, in this case, I'm actually studying how the tumor microenvironment is shaping them and specifically the tumor cells themselves. So I'm actually studying a way in which tumor cells are communicating and this information that they are sending are detected by B cells that
Mm,
can then that my question is how do they respond to this information and can we intervene in the way they respond, can we give something, can we make some type of therapy that would shape the response toward an anti-tumorigenic. And again, B cells are also they mainly be studied in a context of infection, right?
mhm,
Um, but lately, they've been seen to have important roles in cancer. For now, the science is much more correlative. So it's known that B cells might have anti-tumorigenic roles, an increased presence of tertiary lymphoid structures in cancers are usually associated with a better prognosis. But the question is like, why and how?
mhm.
So,
Yeah, the simplest questions are typically the hardest ones to answer.
yeah.
Yeah. Oh, interesting. Yeah, maybe you can share some more details on your current work, and I can include that in the show notes for listeners who are curious afterwards.
Yeah.
I'm switching gears now to the broader story, the broader journey as a scientist. I'm curious. There were probably not a lot. I mean, there were probably a lot of highlights as well as lowlights throughout this journey, and I'm curious what skills, mindsets, ways of seeing the work really influenced you or led to your discoveries or allowed you to persist. Like you can also think of, speaking to a younger version of yourself. What advice would you give yourself?
Yeah. It's tough.
Yeah.
Science is tough because, usually if you're in science it's because you're passionate about that and you really want to find answer to your question, but you usually get more questions and then a lot of time you might feel stuck or lost. I think for me the lowlights were mainly when I felt like, oh, I'm not going anywhere, or like this project is not leading anywhere, or I'm
Mm,
not really finding anything, which is a part that always happen, especially at the beginning of a project, because it's
mm,
normal, I mean, the picture is big, you don't really see. So I would say, I don't know how you can persist. For me, sometimes it was helpful, it's still helpful to maybe take a step back and maybe focus your attention more on either another project or also other things in the lab that might give you joy,
mhm,
like training someone or going to a conference or doing something with your colleagues. And I also find really important sharing your science because I think that's when you share your science with your peers that are maybe not so much into the project, they can easier and faster detect, you know, help you direct the project, help you identify which are the roads, let's say.
mm.
So, yeah,
Yeah.
always ask for advice, discuss. Don't take anything personally. People will comment. I mean, but that's why we are up there, right? To actually
Yeah.
discuss our science.
Yeah, totally. That's a great segue into my next question, which was in this work, you were highly collaborative. And I'm curious what you learned about yourself and others through what had to be a lot of interpersonal communication and negotiation. And how do you think about that in your current work?
Yeah, that's really an important point. I think science, especially with advancing of technologies, it needs to be highly collaborative.
Mm-hmm.
You cannot have all the expertise. You need to ask people and you need to trust people. Um, I think, when you propose an interesting project to someone, nobody will close a door in front of your face if you're like just asking or just even wanting to share your like findings and then maybe asking for advices. Uh, it's definitely, you need to understand and learn how to work with different type of people. The type of knowledge you have to share is different. Talking with oncologists, their goal is obviously different than your goal in the lab, right? But
Mm-hmm.
then the overarching goal is the same. So you need to learn how to talk with everybody, but it's interesting. I find it also fun. I like to put myself in, always in different condition because you keep learning from everybody. Um, I remember one time I have this surgeon that would show me and explain me like all the size of the like colon and stuff, and he gave me like a lecture basically, like an anatomical lecture, and I was like, okay, I just need to know that, but like, okay,
Yeah, that's the flip side of asking for help. Sometimes you get more than you bargain for.
yeah.
Uh, it resonates with my own experience. I think it's really hard to ignore genuine curiosity, and it sounds like that's something that you leaned into by asking interesting and hard questions and being open to the critique and feedback of other scientists. I think when that's rooted in a place of curiosity instead of, I guess, a desire to be correct, you learn more.
Yeah, that's really an important point. I think that's one of the main points of science. You, I mean, you need to be curious. Yeah, and it's just really out of passion and out of trying to understand, trying to like make new pieces of knowledge that maybe today they mean something, tomorrow they might mean something else because there would be more knowledge that,
Yeah.
like in addition to what we know already. And also regarding the collaboration, I think showing to be proactive
Mm.
and showing that you care of what you're doing, that you respect what other people do, and not expecting everybody to be ready to work with or for you, but like showing your interest. I think it's really an effective way to build collaboration.
Excellent point. So, what are you looking forward to the most right now? What gets you excited about your field of study, and how do you see your future vision or goals?
Um, I mean, I think right now what makes me the most excited is I'm really excited about this field of B-cell immunology because I can see like changes at a fast speed, and I'm excited on understanding how they actually behave, and I could see that they could lead to potential new therapies or maybe to like improvement of current therapies, you know. And so I'm excited to see what in the like, I think in the next decade we will have like a new breakthrough in this field. And then it's genuinely, as we just said, the idea of bringing new knowledge in the field that it excites me a lot.
Mm. Yeah. Yeah. Thanks for sharing. We're getting to the end here. And so I wanted to give you just some space to maybe put a message out there. Maybe it's a call to action for other scientists or immunologists, or maybe it's speaking to certain listeners that you might want to connect with who should reach out to you. What are your thoughts?
Yeah, so anybody that is studying B-cell immunology, come to me. I'd love to know what you're doing, to learn from you, to share what we do. Um, and yeah, I would just say, you know, science is a difficult word. Um, we should all do it together. We should have like a type of science that is definitely collaborative, that it's supportive from like a bigger goal. It's not just finding your small answers, but it's really like, oh, let's build collaborative projects that might lead, not now, but in like years, to something that it's really impactful. And yeah, this.
Thank you. Yeah. Dr. Lattanzi, thank you so much for taking the time to speak with me today. It's been a pleasure having you on the show.
Thank you. Thank you so much for the invitation again.
You're welcome. ModernVivo designs best-in-class literature review software to help preclinical scientists identify insights from millions of peer-reviewed papers to design their in vivo studies faster. The ModernVivo podcast was written and produced by Andrés Velarde in the Capitol Hill neighborhood of Seattle, Washington. For more information about our guests, please refer to our show notes. And if you have an interesting story to tell, an interesting science to talk about, reach out to Andrés at modernvivo.com to learn how you can be a guest on our show. Thanks for listening. We'll see you next time.